Hello friends, welcome to BioBrat. In this video, we will be discussing about vaccines, and under the vaccines, we will be studying the live attenuated vaccines and inactivated vaccines. So what are live attenuated vaccines? First we need to understand the word attenuated here. So what does attenuation mean? In attenuation, we decrease the pathogenicity. Okay, so what we are doing, we are taking the pathogen and growing them in such a condition that they undergo the mutation in their genes. So we are growing in unfavorable conditions. Okay, we are growing them in unfavorable condition, which is leading the pathogen to undergo gene mutation. Okay, and due to this gene mutation, they lose their pathogenicity. Okay, so because of the gene mutation and because of growing them in the unfavorable condition, they lose their pathogenicity. That is, their effect is totally decreased. They lose their pathogenicity, but when they enter into the organism, they are able to produce the immunogenic effect because our cells will recognize them as a foreign particles. Okay, so for them, but still, see, this is a pathogen. Suppose, okay, they have lost the pathogenicity. Suppose they're having a gene in their DNA, it has undergone the gene mutation. Now this has lost its pathogenicity, but it will still have the epitope, right? It is still having the epitope, and our immune cells will recognize this epitope, and this will produce the immunogenic effect in our body. Okay, so they have a disadvantage also. So what is the disadvantage? That these the you see here, this has undergone mutation, but it can revert back to its virulence. So disadvantage is it can revert back to its virulence. Okay, but nowadays there has been the improvement in a technology where we can do something the do something where we can stop that mutation back to the virulence. Okay, now this gene has undergone, suppose this is a gene for pathogenicity, this has lost its virulence now. Okay, now this can again turn back to virulence when injected into human beings, suppose. So what we can do now, we can, side directed genetic engineering we can do and stop these genes from reverting back to virulence. Okay, so this is all about the live attenuated vaccines. Now we will be, okay, I'm sorry to, and there is also an example of a live attenuated vaccine. Example of live attenuated vaccine is Sabin polio vaccine. Okay, so this is an example of attenuated vaccine. There, in this vaccine, they attenuated three strains of poliovirus. Three strains of poliovirus were attenuated. Attenuated means they were decreased totally in their effect, the capacity for producing any effect. Okay, these when are introduced into the human body, they introduce the production of IgA antibodies. Okay, when these antibodies are produced, they are present already within our body. So whenever a foreign like naturally the poliovirus is going to enter our body, we already have IgA antibodies for those the virus, poliovirus. So this serves as a defense mechanism towards the naturally acquired poliovirus. Okay, so next we will be studying about the inactivated vaccines. So what happens in the case of inactivated vaccines? In inactivated vaccine, what we do, we totally inactivate the pathogenic organism. Okay, suppose it's a pathogenic organism, we inactivate it totally. Okay, we how do we inactivate these pathogens? We inactivate them by heat or by chemical reagents. Okay, this by these two methods we can inactivate these pathogens. But there is a problem with heat treatment. What happens, we know that pathogen is having an epitope here. So when we do the treatment with heat, the structure of epitope may be lost. They may because heat causes the destruction in the protein structure. Okay, so this may also alter the epitope structure. Agar epitope structure is only not present, how will our immune cells recognize this epitope and produce memory cells? Okay, so what we commonly do in labs is we inactivate them by using the chemical reagents. Some of the chemical reagents used in inactivating the pathogens are formaldehyde and alkylating agents. Okay, so these formaldehyde and alkylating reagents, they inactivate this pathogen totally, and now this inactivated pathogen, these are unable to multiply in the host, but they are able to produce the memory cells within our body. So because they're unable to multiply, there is always a need to provide a booster in these cells. Okay, need boosters. Okay, whereas in the case of live attenuated vaccines, they do not need any boosters. Why? Because these live attenuated vaccines are able to multiply. Okay, these cells are able to multiply. That's the reason they do not need any boosters. Whereas in the case of inactivate vaccines, they are unable to replicate and produce more offsprings. That is the reason it requires boosters. Okay, so this is all about the live attenuated vaccines and inactivated vaccines. So if you have any doubts, please ask in the comment box, and if you like the video, please like the video and subscribe to my channel for further knowledge. Thank you.